Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Quick note before we begin, the Finding Genius Foundation, as part of the Finding Genius podcast, has recently completed a book about understanding viruses. So the creation of this book was to interview 100 virologists, ask them a lot of deep, difficult questions, take the most difficult questions, and then re-interview the top 25 or so and ask them the hardest questions I could think of. And we compiled that all into a book. So you'll see question and 4 or 5 experts' answers. Question, 4 or 5 experts' answers. There's about 30 questions in the book. I think it's a great read for the layperson and for the researcher. It talks about a lot of speculation in the world of viruses, such as are they alive or not, and why is it important? Why do viruses go latent or hidden or ineffective or sit in a person or an animal or another creature for weeks, months, years, and then suddenly become virulent and affect that person? So there's a lot of really provocative questions in the book. It's now on Amazon. So if you go to Amazon and type in Finding Genius, you'll see the book on viruses. It's also on Kindle. The Audible version is in production and should be ready in approximately a month. But if you want to go and order it now, you can do so again by going to Amazon or Kindle, or go to findinggeniusfoundation.org and go to publications. There's an opportunity as well to get the transcripts of all the interviews and to hear the original interviews themselves. If we put them all together, the book would be about 1,000 pages, but we condensed them down to make it juicy and concise and tight and very interesting. So I hope you'll check out the book. We're now working on one about cancer, but this is going to be our goal is 3 times a year to come out with these masterclass books that I think will inspire new scientific research, and I hope you'll check it out. Thank you.
Hello, this is Richard Jacobs with the Finding Genius podcast, now part of the Finding Genius Foundation. I have 2 guests today. I have William Wuest. He's a GRA Distinguished Investigator and Associate Professor at Emory. And I have Kevin Minbiole. He's also a professor and chair, but at Villanova. And we're going to talk about creating natural cleaning products. So both of you, thank you for coming.
Thanks for having us.
Yeah, so tell me a bit about your background and how you got into natural cleaning products. I guess, Kevin, we'll start with you. Go ahead.
Great. Well, Bill and I go back kind of a lot of years. We've been working together for 10 years. We both got our PhDs from the University of Pennsylvania in a group that really focuses on natural product total synthesis. So we've cared for a long time about biologically active compound. And at this point in our careers, we do both mimicking of what nature does and some strictly synthetic compounds, but all aiming towards things with biological relevance.
Okay. And William?
Yeah, and my lab focuses on developing new antibiotics that are potentially more specific toward different pathogenic bacteria, so the bad bacteria that make most people sick. And it was through our kind of a joining of timing where both Kevin and I moved to Philadelphia around the same time as me to start my independent career and Kevin to move to Villanova. And we teamed up, had a meeting together and discovered some of the work that we were doing could overlap in our interests. And that's where we got into creating these new disinfectant compounds that to some extent were inspired originally by these polyamines in nature.
Okay, well, what situations do you guys identify that you wanted to make cleaning products for and why?
That's a great question. We realized maybe 10, 15 years ago that the world's pretty dependent on quaternary ammonium compounds. So these are compounds that you find just all over the place in things like Lysol wipes and just all sorts of other household cleaning agents. They work well, they're not too toxic, and they just pretty much are able to disrupt bacterial cell membrane. So not unlike how soaps work and a lot of other natural products like that. The funny thing is the active ingredients in Lysol were identified in the 1930s. So we realized that there might be some opportunity to improve on some of these chemical structures, especially since nature has been faced with these compounds for a long time. So since the chemical industry makes tons and tons of these compounds and they go off into wastewater, bacteria have been able to spend some time developing resistance to these compounds. So that plus a lack of innovation meant that there was this little subfield just waiting for some creativity.
Okay. What specific applications, again, are you looking at with your products? Are they for, hospital use or personal use or where?
Yeah, I think that's a good question. That was something when we started realizing that what we were making in the labs, actually primarily with undergraduates in Kevin's lab, that we actually had a lucrative market application. So originally these were, I would say, more academic endeavors. And then we realized that we had some new compounds that were pretty good. And we started a startup company in Philadelphia named Novalice Biosolutions to commercialize these things. And one of the first questions we were asked is, what should we apply them to? Because we thought they'd be effective against lots of things. And really what we kind of honed in on was hospital situations where there's actually become resistance to these types of bacteria, or resistance to these types of compounds, these disinfectants that are not being used a lot. And what I think, looking back on it, is a little bit kind of funny, is I think we were 5 years ahead of our time, as now everyone's using these compounds, spraying them everywhere, likely pre-selecting for more resistance with the pandemic that we've been going through for the past year. So now we've reinvigorated this work with a new focus toward potentially applications toward COVID and other things, because they're being used so widely.
Yeah, what do you think is going to happen with this massive overuse of the same compounds over and over and over? You mentioned it very briefly, selecting for bacteria that are resistant, but what do you think has happened already?
Yeah, I mean, I worked for a postdoc advisor who was, I would say, was a discoverer of vancomycin resistance, and he always told us, if you're working in the antibiotic field, you never have to worry about losing your job because bacteria will always become resistant. And when I got into this area working with Kevin, I was convinced that there was no way bacteria could resist something that basically blew up their cells. And that'd be a very difficult thing to overcome. But sure enough, if you look hard enough, you find it. And now I am starting to get genuinely worried that this actually might be something that we have to consider. And fortunately, a lot of our compounds right now don't have similar liabilities as we've seen. That doesn't mean that they're going to be perfect forever. But at least we know we have a little bit more time to keep going if need be.
Well, how are your compounds different from the quaternary ammonium compounds? And what can you say about them? The mechanism of action, or how are they different?
That's a great question. So the mechanism of action might be pretty similar. So structurally, and not to get too chemistry on you or your listener, we build amphiphils. So they've got a polar end and a non-polar end of a molecule, and that allows them to go to the surface of bacterial cell membrane, insert the non-polar end, and disrupt that. Well, things like the active ingredients in Lysol, so benzalkonium chloride, have a single plus charge on them. What we've been doing is building compounds that have 2 or 3 plus charges, which seem to do an excellent job attaching themselves to the bacterial cell membrane and lysing them open. And it doesn't seem to matter too much if it's a resistant strain, maybe a MRSA strain, or it's also really effective against biofilms, which are an entirely different level of complication of pathogenic bacteria.
Yeah, that's what I was going to ask you. Is it a win just to prevent the formation of biofilms or stop them or break them up? Or do you need to go further and actually kill the bacteria that make them up?
Yeah, this is a question this is where my lab started out. But we were very interested in the biofilm activity, and it was always something that I was curious, was it biofilm specific or not? And what our research has, I think, shown is that basically our compounds are just really good at killing bacteria. And bacteria are really good at forming these protective biofilms, and they just become essentially 1,000 times worse against that predisposed environment. But even so, our compounds are still pretty darn effective against eradicating biofilms that are established on these surfaces. So like the pink ring you might see in your sink or on your toilet, or the yellow film that you might see in your shower.
What have you discovered about them that makes them work? And again, if we're getting into any proprietary things, just let me know. But, was there anything different? You said there's more positive charges, so I guess that's more oxidizing potential of the cell membrane, and that's what blows it apart, or what makes it work?
Yeah, I mean, I guess from the proprietary standpoint, and I guess we should have disclosures for this, is that these are patented. These are compounds that we tried to commercialize and are still looking to commercialize going forward. The analogy I use is kind of like Velcro. So the commercially available compounds that are used widely, I would say, are like worn out Velcro. They stick, but they don't stick really, really tightly. Our compounds with the extra positive charges stick really, really tightly to these negatively charged cell membranes of bacteria. And because they can stay there very, very stickiness and stay localized, they're less prone to this resistance development mechanism that bacteria have, and also make them just more potent compounds because they have higher affinity and localization.
What about gram-negative versus gram-positive bacteria? Any difference in action that you need?
Our compounds have pretty uniform activity against the gram-negatives or the gram-positives, which is why we're pretty excited about it. Typical antiseptics really diminish in their activity against either gram-negative bacteria, which are tougher to kill, or resistant bacteria. Most of our best compounds are uniformly active against gram-negative or positive.
Oh, that's good. So yours are not really selective. They pretty much kill any bacteria in an area. Any chance of making selective ones that would go against the bacteria that you consider to be pathogenic?
Before we continue, I've been personally funding the Finding Genius podcast for 4.5 years now, which has led to 2,700 plus interviews of clinicians, researchers, scientists, CEOs, and other amazing people who are working to advance science and improve our lives and our world. Even though this podcast gets 100,000 plus downloads a month, we need your help to reach hundreds of thousands more worldwide. Please visit findinggeniuspodcast.com and click on Support Us. We have 3 levels of membership from $10 to $49 a month, including perks such as the ability to see ahead in our interview calendar and ask questions of upcoming guests, transcripts of podcasts you're interested in, the ability to request specific topics or guests, and more. Visit findinggeniuspodcast.com and click Support Us today. Now back to the show.
Yeah, I think it's kind of a conundrum that my group faces is we are hallmark ourselves on focusing on these specific ones. And then here we are making these broad spectrum, kill everything type of compounds. And what I think about is it comes down to application and use and why you're using it. And a lot of times if you're in a hospital, it's okay. Like you'd rather have a clean, sterile environment and it doesn't need to be as discriminatory because it's not, affecting your microbiome or your gut or things like that. In your household, maybe you'd be a little more selective. Maybe you don't need to use your disinfectants all the time. Maybe you can use soap to clean your countertops or, save the hard-hitting disinfectants for the really tough cleanup so you're not, again, predisposing the environment to these compounds and pre-selecting for resistance. But I would say in the antiseptic fields, they would much rather something that killed everything and sterilized as opposed to something that was selectively killing pathogens.
Yeah, but what happens if the compound, people touch it, which I'm sure they inadvertently do, with or without gloves, but I'm sure they would eat little bits of it. They would volatilize a little bit. They would inhale bits of it, etc. Bacteria that are, we'll just stick with that. But at what point do you test how people are affected by it that use the cleaners or in the presence of them?
Yeah, part of our research does focus on those aspects. I should note that there's a well-known quaternary ammonium compound called cetylpyridinium chloride that's in a lot of mouthwashes and already, I would say maybe not intentionally digested, but exposed to it. And some of the studies in our lab look at the toxicity or therapeutic index of our compounds to see if we have selectivity. And we've been lucky to find some that are selective and slightly less toxic than the commercial compounds as well. But it is definitely a battle you face as human membranes or similar to bacterial membranes. So the mechanism would be similar for toxicity in each case. But to be clear, historically, these quaternary ammonium compounds are incredibly safe compounds used broadly with very minimal side effects. So I use them. I stand by them. I think they are very safe compounds to use for these types of applications.
But Rich, you bring up a good point. We are really trying to optimize the ability to kill bacteria and leave human cells untouched. One of our hallmark strategies is just to build a heck of a lot of different compounds. So we're up to almost 700 different compounds that we've built up here at Villanova, and we ship to Bill's lab for him to test, because we're really trying to learn as much as we can about structure, activity, relationships to really optimize what's the best compound at killing the pathogens and leaving eukaryotic cells.
In certain applications, are you able to target a specific microbe, or again, you're finding the general approach is working better? Can you modify this approach to go after targeted microbes?
So I would say we can, but I would say in the class of molecules that we're talking about currently, these quaternary ammonium compounds, that's very difficult. Going back to kind of some of the things you mentioned earlier in terms of natural products, what my group has been focusing on recently is looking for inspiration from nature, bacteria that grow in the soil, to see if they've evolved ways to make specific compounds to kill the pathogens around them. And this has kind of motivated a bunch of different angles in our lab to identify compounds that target, say, specifically Pseudomonas aeruginosa, which is prevalent in cystic fibrosis patients' lungs, or strep mutans, which is found in plaque in teeth that causes cavities and endocarditis. And these are certain spin-off projects that I'd say the compounds are more elaborate and more complicated, but have the potential to be used either prophylactically or in other ways going forward.
In the crazy idea department, one of the ways that we've modified some of the compounds we built is to add iron atoms attached to the structure. While we're a little bit rudimentary right now, this is kind of inspired by some Trojan horse style chemistry, actually from the university that we both graduated from, Notre Dame, in attempts to get the bacteria to try to actively uptake some of these compounds, which would be a great way to, because bacteria need iron to survive. This might be a sort of strategy, if we can refine it, to have bacteria actively take these poisonous compounds towards themselves and lyse them.
What about acting in other ways, interfering with the quorum sensing that bacteria do so that they won't get to the biofilm stage or they won't turn on a certain ability? What else can you target instead of just blowing the membranes open?
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Yeah, it's a very sophisticated question, getting to the quorum sensing and antivirulence type compounds that target what makes bacteria make you sick. And these are approaches we thought about in our lab. I go back and forth on the merits of those because I don't know how you would get such a compound through, say, FDA approval, because you're not killing the bacteria. So the thresholds for what would qualify as an active drug or something that's effective would be a little bit harder to quantify and be able to get through these clinical trials. But there's definitely many research groups out there in academia and industry looking at these creative approaches that might be less prone to resistance.
Well, how do you guys get your ideas? Do you observe bacteria in action in certain environments? Have you looked at, this is one thing I'm curious about. Has anyone been able to swab or look at areas that have been cleaned, 8 million times and see what kind of bacteria are now there before, during, and after cleaning, and look at a reference material, let's say wood, like a wood door or something, and treat one with chemicals, every day, all day, leave the other one alone, or just use, older chemicals like soap or things like that, and see what kind of bacteria develop on both. And then you might have a better strategy on who's there when you need to clean it after it's been cleaned a million times.
Yeah, I mean, there's definitely people investigating, looking at, selecting for resistant microbes and things like that. I would say, from going back to your first question about where do we come up with our ideas, at least for me, I like playing detective. I like looking back into older publications, looking at environments where there's pre-selection for compounds that might kill bacteria. So again, going back to my analogy earlier, looking at the soil around plants, there's lots of microbes in the plant soil that are trying to kill each other and compete for their viability in the areas. So looking back at the bacteria that grow in those areas, you might find compounds discovered in the 30s, the 40s, the 50s, the 60s that were overlooked. Maybe they weren't effective at being anti-cancer compounds that they were searched for, or other reasons, and weren't looked at as antibiotics. And these kind of looking at the structures and looking at where they were found can kind of give you hints that you might be finding the next great thing. For antiseptics, it's tricky, right? It's really hard to find a compound that you can produce for incredibly cheap prices and commercialize in large amounts of material that is both safe and broadly applicable. So it's a tricky battle of that balance of activity and cost and safety. But it's something that motivates our work going forward.
Well, what's observed in nature? Do bacteria typically, when they make antimicrobial peptides, do they always seem to target the exterior membrane to lyse it, or do they have other methods of action that you could, make compounds to mimic?
Yeah, so antimicrobial peptides are a great example. It was, I think, the motivation to one of our first papers in these class of molecules is kind of trying to form simpler versions of that. And what's amazing about those molecules, these AMPs, is that they are incredibly selective and not very toxic to human cells, which is really amazing and remarkable. And I think many companies have been developed around that idea, or using them for polymer coatings or other work. And what other compounds are out there? I mean, there's lots of other main targeting molecules. Some target the ribosome, which helps bacteria make proteins, target other types of mechanisms of viability in life. And it's really remarkable how many unique targets there are out there. And we keep finding new ones in our lab through the research that we're doing. And really the goal is to try and find those targets that are only present in pathogenic bacteria. So then you really would have something that's very specific. And again, it's keep looking and getting motivated about what nature has been doing for millions and millions of years.
It's incredible how ubiquitous these antimicrobial peptides are. I mean, to my understanding, virtually every organism that's not single-celled makes antimicrobial peptides in an attempt just to keep out the bad guys, which again are just ampliphiles trying to, generally speaking, lyse bacteria that are coming by.
So the method of action usually is lysing their competitors, lysing their cell membranes, but not other methods of action.
So for the antimicrobial peptides, that's correct. But there are other compounds that these bacteria can produce that target different things.
Okay. What's commonly seen? Let's say again, I have a table in a restaurant, and it gets cleaned, after every meal, et cetera. What would you expect to see there Like, what are the bacteria eating versus bacteria that would be on us or in us? What would be like a common food for them that would cause them to, to go there and hang out there? What are they eating?
I would say one of the common things that if you're growing bacteria like we do in our lab is basically the carbon source. So whether it's sugars that are left over, residual syrup that might be on the table, or other kinds of carbon sources that the bacteria can then break down, digest, and use for their energy production. Typically, it's they need different types of ions, salts, and sugars to keep their process going. But I should note, a lot of bacteria actually are really, really good at surviving in really harsh conditions. So whether if there's starvation of materials, they will form these biofilms, or some bacteria will form spores to protect themselves from others. And they're really phenomenally adaptive at surviving in really harsh, unique conditions and areas.
Oh, so a lot of times they could be on a surface where they're getting no nutrients, but they're in a spore or a biofilm form, waiting for some contact from some organism, and off they go. Now they have access to salts and sugars and all kinds of good stuff.
Exactly, and that's where quorum sensing comes into play, where they can maybe come back and say, "Hey guys, we have more food. Let's grow more. Let's reproduce and expand, and maybe venture out for new places to live."
I don't know how crazy this is, but has anyone made a dry soap, or could you actually, compress soap and make a doorknob out of it, so that every time someone, grabs the doorknob, maybe a tiny bit of the oils in their hand goes into the soap and activates it and kills stuff on the surface?
It's not crazy to look at surface disinfection. You can attach antimicrobial compounds to surfaces, and that can, I think, happen with quaternary ammonium compounds or a variety of other things.
Okay, I just thought it was a different idea. So what have you found in nature that really, I don't know, maybe you haven't been able to mimic it yet, but just really amazes you? I mean, one thing you mentioned is there's antimicrobial peptides made by all kinds of creatures everywhere, but in your exploration, what other really interesting or fascinating things have you found?
So there's one story in my lab that we've been working on for the past 5 years or so, where we were interested in this small molecule made by a bacteria called Pseudomonas, and it was a relative species to Pseudomonas aeruginosa, the one that I mentioned earlier about cystic fibrosis and other wound injuries. And what we found that was interesting was this one bacteria makes a compound that specifically only killed the bad guy, Pseudomonas aeruginosa, and really nothing else. So we were really intrigued by this. No one's ever made it. No one understood how it worked and if it was a legitimate finding. So our group made it. We made some compounds that looked similar and simpler, and we figured out how it worked. And what was really, I thought, remarkably surprising is that it targeted an enzyme in primary metabolism. So some enzyme that every bacteria, even humans, had. But it was so specific in either how it's getting into the bacteria or on how the enzyme looks in bacteria compared to others, that it really only targeted that one species. And this is something that we're hopeful that we can move further and look in using it into maybe a new antibiotic in the future.
Are there any bacteria that you could culture and put into a spray, and they would act as the disinfectant because they would, phagocytose other bacteria or just attack them when they're near?
That's another great question. A long time ago, when I worked at James Madison University, I started teaming up with some ecologists, salamander ecologists, who were looking at the bacteria on salamander skin and how they could fight off fungal. So I ended up looking at a lot of the chemicals there, which was kind of a fun story. Some of that technology has since moved on to a startup company that looks to use probiotic bacteria. So bacteria you purposefully apply to human skin to fight off human fungal infection. So the application of good bacteria doesn't have to just be in yogurt that's going through your digestive system. It can be actively applied to your skin to keep the right symbiotic balance of microorganisms on your skin.
Are there any fungal or mushroom-based cleaners? Because fungi seem to have really interesting defenses to bacteria and vice versa.
I'm sure there probably are none that come to mind specifically, but I agree with you. There's definitely compounds that are produced by fungi that have been investigated for antibacterial activity. I know there's also some really interesting research in bacteriophages. So these really small phages that basically target bacteria specifically and kill them as well. And I think down in outside of Atlanta, there's an institute looking specifically at bacteriophages as a new approach for antibacterial treatment.
Yeah, I guess there's tons of angles you can go. Look at in an ecosystem, what's interacting right now. Is your bacteria that you're trying to target in a certain ecosystem? Who's killing it in that ecosystem or fighting against it and co-opting their abilities to be used in other applications is the name of the game, it sounds like.
You're describing the field of chemical ecology. I mean, it's just chemical warfare from these little microorganisms that can't run away from each other or attack each other. It's all chemical warfare at a molecular level.
So what other projects are you guys working on that you're hoping to, bring to the market in the near future?
Well, I'm still working with that skin probiotic company called DermBiont. We're trying to see what are the best symbiotic bacteria that you can apply to humans. How do they fight off different fungal diseases, and what chemicals are active in the situation?
And we're kind of playing again the game of being Sherlock Holmes, digging through literature and trying to find the next, newest, greatest molecules that could be specific in their approach and maybe be used again for certain angles that having a very narrow spectrum type of compound would be beneficial.
All right. And what do you guys expect to be able to produce in the next, couple of years? What's like real near term that you're very close to having a breakthrough on?
Well, I think that we've developed hundreds of promising new antiseptics, and it's just going to be a question of which one is going to be the best way to balance efficacy versus safety. To circle back to a question you had said before, if we're making tons and tons of these antiseptics that are going out into nature, how can we do better? Well, the answer is have more effective compounds that don't really get slowed down by resistant bacteria, and maybe we can use them in much 10x smaller amounts to still get the job done and clean that hospital room. So in the short couple year term, that's what we're working on between the two of us.
Yeah. What about a cocktail of, known chemicals that'll work? But if, let's say you have 5 of them, you need hopefully 1/5 the amount of each. Would something like that work better?
Yeah. I mean, that's something that I've been really thinking deeply about. I kind of looked to HIV therapies as motivation. The HIV cocktails have been incredibly lucrative and effective for really almost eradicating the disease in some ways. And I think there's a lot of things that can be learned there in terms of combining different agents with different mechanisms of action to make it very hard for bacteria to overcome that resistance profile they could possibly develop with one compound being used.
Well, very good. What's the best way for people to find out more about your work, both of you? Where can they go?
So for me, I have a website at Emory that if you search my name, you'd be able to find. I'm also pretty active on social media on Twitter at WMWeast, WUEST. And we typically post our latest and greatest there as well.
And for me, head to the Villanova Chemistry website. And if you can pronounce or even spell my last name, I'm pretty easy on a Google search.
Yeah, your name looks biological. Minbiole. It seems like a structure of a bacteria or something.
Yeah, we try to minimize. No, I have no idea.
Well, very good. Well, both of you, thank you for coming. And you're both working on super interesting things from different perspectives. And I think it's great. So thank you.
Hey, thanks so much for letting us talk a little bit with you.
Thanks for having us.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.